Good morning. It's Wednesday. Wednesday's garbage day. Better make sure those garbage cans got out there. Maybe it's not for you. Anyway, here we are. Quiz 3. So this had some stuff that you guys weren't used to, so I'm going to go over it. And once again, we're learning test taking skills as well as flexing our biotech muscles. Which of the following is not considered a significant advance in PCR methodology? Methodology means like how it's done. Yeah. So the invention of the thermal cycler, that's pretty sweet because before you used to have to take the tube and put it into 3 different temperature water baths manually over the course of hours. That was horrible. So this is sweet. The use of reverse transcriptase to generate cDNA, that's complementary DNA from single-stranded RNA. That's how the PCR test for coronavirus works, and that's relatively new, and it made PCR sweeter. The use of agarose gel electrophoresis to visualize amplified products. We already used gels to see DNA. In fact, PCR could not have been invented if gel electrophoresis hadn't previously been invented. So this came before PCR. This didn't come after PCR. So this is the right answer. Incidentally, the use of fluorescently tagged probes for real-time quantitation, that's measurement of PCR products. That's that's real-time PCR, and that's how the coronavirus tests works also. So the coronavirus test does reverse transcriptase PCR, and it does real-time PCR. Okay, enzymes have a wide range of applications in research. Match the enzyme to its potential use in the laboratory. DNA ligase, that's the one that glues the sugar phosphate backbone together. So there we go. That looks good, joining pieces of DNA together. Heat stable DNA polymerase. So our DNA polymerase in our bodies wouldn't work for PCR because it wouldn't be able to stand those near boiling temperatures. So that's why we use the TAC polymerase, the Thermus aquaticus polymerase, in order to perform PCR reactions. Sweet. Protease. Maybe you never heard of this before. It's okay, because you know that ACE is an enzyme that works, and this one works on oh yeah, proteins. So it must be this one, degrading or breaking down proteins. And restriction enzymes, you know that we use those to do restriction digests. So we can see right here that we get digesting DNA as choice. Booyah. Question 3, it's debated that genetically modified food crops have both beneficial and negative effects on humans and the environment. Which of the following is a potential negative outcome of growing or consuming genetically modified crops? Choose all that apply. The nutritional value of a genetically modified crop may increase. Well, if that's true, it wouldn't be negative. It would be positive if the food was, more nutritious. Genetically modified crops may cross-pollinate with non-genetically modified crops. You may have heard that this can and does happen sometimes, and that is a negative. You don't want your GMO DNA getting into the organic farmers' genetics next door. Growing only genetically modified crops may decrease genetic diversity. That would be a bad thing, and sure, that sounds pretty possible to me. Genetically modified crops may be more hardy, like able to deal with heat and drought. That would be a good thing, not a bad thing. So we're not going to choose that because they were talking about negative outcomes. Question 4, which of the following is an advantage of treating diabetic patients with genetically engineered human insulin rather than cow or pig insulin? So they used to grind up pig pancreases to get insulin, and then they'd purify it and inject purified pig insulin into type 1 diabetics. And that worked better than not working, but it wasn't as killer as recombinant human insulin. Anyway, which of the following is an advantage to recombinant human insulin? There's less of a chance of an immune response to human insulin than pig insulin. That's true. You're not going to get an allergic reaction because it's from humans. Ethical issues concerning the use of animals are avoided. Yeah, you don't have to grow a bunch of pigs and cut out their pancreas. So that one sounds good. Oh man, it deselected this. Oh, look, we got all the above down here. Let's just check. It is a more efficient process to purify insulin from yeast or bacteria than it is from animal organs. That seems reasonable to me. So yeah, I'm going to go ahead and click all the above. Bioethics is best described as, you don't need to have ever heard this word before. Bio, you mean that refers to life. And ethics, I hope you understand that means the study of what is right and wrong. So bioethics, the study of decision making as it applies to moral decisions that must be made due to advances in biology and technology. Like, just because we can make mice glow green, is that something we should do? That's bioethics, and that's what we're talking about. We think it's this one. We better test taking skills, better read these ones. The evolution of better adapted organisms which do not destroy more primitive forms. That's an idea and that's something, but that's not bioethics. The portion of the brain, no. The study of biology as it relates to changes in society. Now, this is a less good answer than this one up here. If this one wasn't here, then maybe I would choose this one, but this one's sweeter, so I'm choosing this one. It's the best answer. Why might some people be ethically, again, ethics, opposed to using CRISPR-Cas9 to create mutations in animal embryos? So this is like that guy in the video we were watching, Unnatural Selection, the guy with the dogs and stuff. He wanted to make these like gnarly dogs or whatever. So why might some people not be into that? It says, alterations in somatic cells will be passed on to future generations. And then alterations in germline. Somatic germline, what's that? Here's what that is. You might remember from biology class that if you have a mutation in your somatic cells, like for example, this Giwa has a mutation in some cells in her breast that leads to breast cancer, she could get breast cancer, but that doesn't mean her kids are going to get breast cancer because babies aren't made from breast cells. That's as opposed to her ovary right here. If she got a mutation in a cell in her ovary that produces eggs, babies are made from eggs and sperm. So that mutation could be passed to this child, all cells in the child, because all cells in the child come from this one starting egg cell right here, combined with the starting sperm cell. So if you have a germline mutation or a germline edit that was caused by CRISPR-Cas9, then that's going to be passed on to your children and your children's children and your children's children's children, etc. And that's a big deal. So that's what we're talking about right here. Alterations in germline cells will not? No. Alterations in germline cells will be passed on to future generations, and that's an ethical consideration. Like, what if I want a huge butt? And so I use CRISPR-Cas9 to give myself a huge butt. That's cool for me. But if it's in my germline cells, then all my children and children's 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 are going to have huge butts too. And maybe they don't want huge butts. That's not cool. I have to leave it up to them. And then they could just do squats. Match the person or group with the best example of their responsibility in a biotechnology company. Okay. This one I was a little concerned about you guys right here, but you're going to be sweet because I'm going it over with you. Ensure that all government regulations are followed, working closely with all departments. Let's see. Quality control, wait, man, these are like the same thing. What the hell? Here, let's check it out. Quality assurance, like if we did a bubble chart or something, quality control falls under quality assurance. You guys know how when we were having problems in the lab and I was saying, you guys know how I'm a little bit railer and I don't keep things that nice. Like, well, quality assurance are people that have the job of making sure that the lab is running killer so that the products of the lab are killer. And that's everything. That could involve hiring the right people, that could involve training, that could involve looking at the workflow to look for possible sources of error, that could be managing equipment to make sure that it's operating properly. These people are doing lots of different stuff. That's quality assurance. And a department within quality assurance is quality control. And all quality control does is two things. When raw materials come in, like EDTA or DNA ligase or Taq polymerase or something, they test, they do physical tests on those ingredients, reagents, to make sure that they work. Then, once the product of the lab is produced, they test the product of the lab in order to make sure that it does what it's supposed to do. And then they report their findings to quality assurance. Quality assurance are the bosses of quality control. So let's go back and see how we do with this. Match the person or group with the best example of their responsibility in a biotech company. Ensure that all government regulations are followed, working closely with all departments. That's like a lab manager. That's quality assurance. Conducts tests on raw materials, in-process samples, and finished products. That is quality control. Participates in the production of commercial quantities of a product. So these are the people cranking out, like the pirate plasmids, for example. You guys were working in manufacturing when you were doing that. When you tested it, you were doing quality control. I was doing a crappy job of quality assurance. Works to identify new or improved products or processes. That's research and development. That's the real, like, nimble brains of the lab. That's the people that sort of think outside the box. That's research and development. They're coming up with new product ideas. Which of the following is the study of the entire set of proteins of a cell or organism? Study of proteins. That would be proteinomics. Okay. Which of the following is the study of the full complement of DNA in a cell? That means the full buttload of DNA in a cell. That would be genomics, the study of the genome, which is all of the genes in an organism. Question 10, the major step in the production of a product is listed below. Place the steps in the correct chronological order. Okay, we're trying to make a product. What do we need to do first? Quality control tests and approves or rejects raw materials following test records. That sounds like it could be first. Quality assurance approves or rejects the final product. We're already talking about final product. That's probably going to be the last one. Quality control tests the final product. We're talking about the final product. Manufacturing produces the product. That's going to be, all of that's going to be after quality control tests and approves or raw materials. That's reagents coming that you bought. So boom, that's going to be first. Next, you're going to have to manufacture the product. Yeah. Next, quality control is going to have to test that sucker. See if it's killer. And then finally, quality assurance gets to look at all those reports that all those other people made, and they're like, yeah, we're going to stamp this sucker, and we're going to put our company's name on it and reputation, and we're going to sell it to consumers. So they get the final say on whether that happens or not. Who's they? Quality assurance. Yes. Question 11, maintaining a safe work environment is critical for the well-being of all employees. Wear your safety glasses. Which of the following agencies is responsible for making sure that you're wearing your safety glasses? That would be OSHA, which stands for Occupational Safety and Health Administration. And every once in a while you're working in a lab, you'll get a heads up from quality assurance. OSHA is coming on Tuesday. We're going to go have a meeting right now to make sure everyone's doing what they're supposed to be doing. Because if OSHA comes in here and sees someone not wearing safety glasses, they're going to write us up and maybe we'll get a fine. OSHA. The U.S. agency responsible for regulating biopharmaceuticals for use in humans. So this is the U.S. government agency regulating biopharmaceuticals. So that's drugs for use in humans. That would not be the Department of Agriculture. That would regulate the use of drugs in animals and plants, non-human animals and plants. The Food and Drug Administration, FDA. Ever heard of it? FDA. FDA approval. Yes? Patenting, it's getting a patent, a new biopharmaceutical will give a company exclusive rights to manufacture the product for a term of 20 years in the United States. So if you design and patent a drug, you get 20 years to sell it exclusively. That means no generics. One reason the US government does this is to incentivize companies to invest in developing new drugs with the security of knowing they might offset all of the costs that they put into developing that drug through revenue, that's cheese, gained by selling the drug exclusively. If you develop it and patent it, you are the only one that gets to sell it for 20 years, and you get to make a bunch of dough before generic companies can start selling the drug that you invented. Sounds fair to me. True. Which department within a biotechnology company is responsible for document control and for the oversight of regulatory compliance? We're doing everything the government tells us to do. That would be quality assurance again, man. They got their hands in everything. You need them. You hate them, but you need them. Good laboratory practice, GLP, is a quality system intended to ensure regulatory compliance in preclinical laboratory studies. All this means, you guys, good laboratory practice is wear gloves, keep your enzymes on ice, change your pipette tips, keep your dirty pipette tip receptacle, keep the lid on it so nasty stuff doesn't get in there and infect things in your lab. That's called good laboratory practice. That's what I've been teaching you guys all year. You know how to do it. You guys are pros at gulp. Match the clinical trial phase with its description. Phase 1, phase 2, phase 3. You guys could figure this out on your own. Good test taking skills. But I'm just going to show you this right here. Clinical trial phases. So here's the dealio. Okay, so on the path of a drug being developed and discovered to it being approved by the Food and Drug Administration, it has to go through clinical trials. That means you have to test it to see if it is safe and to see if it works. So phase 1 of that involves 20 to 100 volunteers, and you just need to check to see if it's going to hurt people or not. And you don't want to check that on 10,000 people, because if it does hurt people, you don't want to hurt 10,000 people. You just want to hurt 20 to 100 people. So you use a small amount of people checking for safety, phase 1. If it's safe, then you can give it to people with the disease to see if it works. And again, you don't want to do it to a whole bunch of patients, maybe 100 to 500 people with the disease, and check to see if it works. Phase 3, you do it to more patients with the disease. That way you can find if there's any outliers and people that are getting sick with a larger sample size, if you're more likely to see an outlier or somebody getting sick. And at that point, it's also compared to existing drugs for the same disease. If this drug you're testing turns out to be not as effective as some drug that's already out there, then FDA is not going to approve it. Phase 4 is when it's already improved and the FDA monitors its effect on people that are taking it once it's in the marketplace. So phase 1 to phase 4, that's the stages in getting a drug approved. Okay, phase 1. Phase with volunteers who have the disease. No. Phase with healthy volunteers or people with the disease condition. The primary focus is on studying, I can't see it. I'm going to have to make the screen smaller so I can see it. Phase with healthy volunteers or people with the disease condition. The primary focus is on studying safety parameters of the drug. Yeah, that's phase 1. Booyah. Safety parameters. Good. Safety first. Yeah. Next, we want to see if it is effective or if it has side effects. So that would be phase with volunteers who have the disease. The primary focus is on efficacy, and that's how effective it is, and determining side effects. That's phase 2. And then phase 3, broad. That means a buttload of people. That's going to be phase 3. You got that? Broad. Should I go back and see that again? The primary focus is on a broad scale study of the drug's efficacy and monitoring side effects. Yes. Okay, question 17. Enzyme maltase, here we go again, catalyzes the breakdown of maltose into glucose. A technician, that's you, add maltase to a solution of maltose in order to be incubated at 37 C butt temperature. Samples were taken from the reaction tube every 5 minutes over the course of an hour to measure the glucose concentration. After an hour, the technician noticed that the bath failed to reach butt temperature. It only reached a little bit warmer in room temperature. What should the technician do? Repeat the experiment again. See if it works that time. Seems reasonable. Report the problem to quality assurance personnel or the individual in charge of equipment repair. That seems reasonable. Report the problem to quality assurance or the individual in charge of repair and place an out of service label on the water bath, or do nothing. This is it right here, man. Tell the boss, hey man, something funny's going on with this sucker, and put an out of service thing on there so someone else doesn't screw up their experiment too in the meantime. It's just being responsible in the lab, looking out for everybody. Which of the following is a controlled document that contains step-by-step instructions on how to perform a routine activity with a company? I call that a protocol. Another word for a protocol is a standard operating procedure. Boom. Yes? Standard operating procedure. Do it like this. It's like a recipe, and you need to follow it when you're working in the lab. Which of the following is a controlled document? Here's another document that contains step-by-step instructions on how to manufacture a product. Sounds like the same thing, including spaces for you to write stuff. That's a batch record. And when you're producing a product, you keep a batch record, and that batch record can go to prove that you did it the way you were supposed to do it. And it can also include data like concentrations and stuff like that, yields. Okay. Why must a company manufacturing a controlled product maintain documentation on all processes and checks? Why do you always need to write everything down in a standardized way? Well, documentation provides objective evidence that the product was made and tested correctly. Yeah. Documentation ensures that all personnel follow the proper procedures. Yes. Documentation ensures that all equipment is operating correctly. Are your pipettes calibrated? Documentation provides a traceable record for FDA when they come to inspect your scene. All the above. Submit quiz. How do we do? Did we get hella right? 23.5. Correct. 23.5 out of 23.5. We totally killed it. I'm sorry that was so long. Take notes, especially on the somatic versus germline part and on the phases. Yeah.